Good morning. Today we are going to discuss the fermentation process, which is a part of biotechnology portion. And in biotechnology portion that we are cover the old biotechnology and new biotechnology model. Okay, the old biotechnology model. In case of old biotechnology model, that involve the whole part of living organism that used by the process called fermentation. That's why we cover the fermentation overall process. Okay. So first we are clear the definition of fermentation process. Fermentation is an anaerobic oxygen-free metabolic process where microorganisms like yeast and bacteria convert carbohydrate into acids, gases or alcohol. Okay. So first of all we are clear that fermentation is an anaerobic metabolic process. Anaerobic means that is absence of oxygen, okay, where microorganism, that can be yeast, that can be bacteria, that can be virus, the different kinds of microbes, okay, that convert carbohydrate inside of carbohydrate that is present starch or sugars, in case that help to processed into acids, gases or alcohol. Okay, so that process is called fermentation process. Fermentation is based upon microbial activity or developmental of microorganism. That based of fermentation is only depend on microbial activity. Okay, the first convinced by Pasteur in 1857. First he discovered the microorganism required for fermentation process. Okay, so first we clear that how the fermentation process are very beneficial to us. That process is glucose that convert into to pyruvic acid. That process is glycolysis process. Okay, that glucose, one molecule of glucose that convert into two molecule of pyruvic acid. That two molecule of pyruvic acid that can convert ethanol, either lactic acid or organic acid, different types of organic acid. Okay, the fermentation process begins with glycolysis. The first of all, the fermentation process begins with the glycolysis process, and glycolysis, what glycolysis, glucose is broken inside of the glycolysis process. The glucose is break down into pyruvic acid molecules, and then the pyruvic acid molecules are converted into ethanols, lactic acid and organic acid. Mostly two types of fermentation are required, that is alcoholic fermentation and lactic acid fermentation. Okay, the first of all, alcoholic fermentation, that is pyruvic acid. Here is the pyruvic acid converted into acetaldehyde, either CO2, carbon dioxide. Okay, so here the microbes are required. Here the microbes are required, that is can be yeast. Okay, in acetaldehyde, pyruvic acid converted into acetaldehyde, that presence of microbes, that examples are yeast. Okay, then acetaldehyde reduced into ethanol, that can be converted into ethanol. Okay, the same process here be repeated. This is carried out by yeast, that is Saccharomyces cerevisiae. So here is the record of yeast, that is Saccharomyces cerevisiae. This process used by the produced bread and beer. Here is useful for bread formation and beer formation, wine formation and spirit formation. Okay, in this process CO2 is removed. Here in this process, that pyruvic acid and acetal converted into acetaldehyde, then the ethanol, that can be reduced or that can be removed CO2, carbon dioxide. Okay, that the carbon dioxide helps to release and help to rise in bread formation. That swollen part of the bread, that can be responsible only the carbon dioxide. Okay, so first of all, pyruvic acid are here is the one part is complete, that is pyruvic acid converted into ethanol. The second part is the pyruvic acid converted into lactic acid. Okay, the pyruvic acid are reduced to lactic acid by the presence of bacteria, that can be bacteria streptococcus and lactobacillus bacteria. Okay, the pyruvic acid is reduced to lactic acid. The pyruvic acid are reduced to lactic acid by presence of bacteria streptococcus. Okay, it is carried out by the bacteria, example streptococcus and other many bacteria, lactobacillus. Okay, it is also used for storing milk for production of a cheese. Here is the purpose of pyruvic acid, why the pyruvic acid is converted into lactic acid. The last is the best definition of fermentation is conclusion part, that is it is chemical transformation. Here is the chemical transformation that can be organic substance converted into simpler compound by the action of enzyme that can be secreted by such as microbes such as yeast, fungi and bacteria. Here is the total view of fermentation process, and next video is the fermenter. What is fermenter and what is the device of fermenter required for fermentation. Thank you.